When you walk into the produce section of a grocery store, there's this illusion of neat, orderly botanical categories.
Absolutely.
You have your leafy greens misted with water over here, you've got your heavy root vegetables stacked over there, and then your cooking oils are bottled up in another aisle entirely. It feels like a perfectly organized, distinct world.
Yeah, we definitely take comfort in putting things in their own little boxes, like a cabbage is a cabbage, a turnip is a turnip, and, mustard is just a condiment for your hot dog.
Exactly. But if you actually pull back the curtain on the world's most versatile family of vegetables, the Brassica family, that orderly grid, completely shatters.
It really does. It's wild.
It is basically a genetic hall of mirrors. That head of broccoli, the turnip, the leafy bok choy, and even the canola oil you fry your eggs in, they might look completely alien to one another, but they share this incredibly intertwined, chaotic ancestral DNA.
And tracing that DNA has historically been an absolute nightmare for plant geneticists. For decades, mapping the relationships between these crops felt like trying to navigate a dense jungle at midnight with a dying flashlight.
A dying flashlight, yeah.
The genetic landscape was just full of blind alleys.
Which brings us to the guiding document for our deep dive today. We're breaking down a monumental 2026 paper published in Nature Genetics by Juncea and colleagues.
It's a fantastic paper.
It really is. It's titled Genomic Analysis of 3,330 Accessions, provides insights into the evolutionary history and self-incompatibility locus of the Brassica A genome.
Quite a mouthful.
Yeah, quite the title. But our mission today is to decode how a literal weed growing in ancient Mesopotamian wheat fields was completely transformed by our ancestors into these wildly different global crops.
And we're also going to look at the underlying mechanics of how these researchers finally unlocked those secrets. Because they used a groundbreaking mapping technique that solves a massive bias problem, a problem that has plagued not just plant genetics, but potentially human genetics as well.
Right, the methodology is just as fascinating as the history. But before we can trace the globe-trotting history of these plants, we really need to understand the architecture of this family tree.
For sure.
In reading through your notes, there is this foundational concept called the triangle of U. Now, I'm picturing a literal geometric triangle, but how does this actually dictate what ends up on our dinner plates? Let's unpack that mechanism.
Well, a literal triangle is actually the best way to visualize it. This model was proposed back in the 1930s by a Korean-Japanese botanist named Woo Jang-chun, who is often referred to as Yu.
Okay.
Picture a triangle, and at the three distinct corners, you have three base diploid species.
Diploid meaning S.
Right. Diploid just means they carry two sets of chromosomes. Similar to humans, we get one set from each parent.
Got it. So what are the corners? Let me guess. One has to be cabbage, right?
Close. The corners are the foundational genomes. So at one corner, you have Brassica rapa, which carries what we call the A genome. This gives us turnips and bok choy.
Okay, A is for turnips and bok choy.
Then at the second corner is Brassica nigra, carrying the B genome, which is black mustard. And the third corner is Brassica oilseed, carrying the C genome, which includes your cabbages and broccoli.
So the A, B, and C genomes are the isolated corners, but where do all the other variations come from, like canola oil?
They come from the sides of the triangle. See, in nature, these corner species sometimes cross-pollinated and literally smash their entire genetic codes together.
Oh, wow.
Yeah, creating tetraploids, which are plants with 4 sets of chromosomes instead of 2.
So they just double up.
Exactly. If the A genome turnip crosses with the C genome cabbage, you get the AC genome, which is Brassica napus, widely known as rapeseed or canola. And if A crosses with B, you get the AB genome, Brassica juncea, which gives us brown mustard.
So the A genome Brassica rapa seems to be the heavy lifter here. It's a corner base, but it's also a foundational building block for both rapeseed and mustard, which makes it the star of this Nature Genetics paper.
It absolutely is.
But if scientists have known about this triangle since the 1930s, why did it take almost a century to map the A genome accurately?
That delay comes down to a methodological blind spot. To study the genetics of any species, researchers traditionally rely on a single reference genome.
Meaning they just pick one plant.
Basically, yeah. They take one individual, in this case, a type of Chinese cabbage called the Chifu genome. They sequence it perfectly, and then they use it as the master blueprint for the entire species.
I mean, that sounds reasonable on paper. If I want to know how a car engine works, I look at the manual for a Honda Civic and assume most other sedans are built roughly the same way.
Right, but the flaw in that logic is that the Brassica family is infinitely more diverse than a lineup of sedans. When researchers recently sequenced thousands of other Brassica varieties and laid their DNA over that single Chifu blueprint, the results were alarming.
How bad was it?
Only 33 to 64% of the whole genome sequences in those other varieties actually matched the reference.
Wait, I need to pause on that math. Are you saying that up to 2/3 of the genetic code in some of these plants was just missing from the official scientific map?
Completely missing. Entire sequences, genes responsible for disease resistance, flavor, climate adaptability, they were completely invisible to science because the reference point was too narrow. If a gene didn't exist in that specific Chinese cabbage, researchers didn't even know to look for it.
It's like trying to understand all of human literature by only studying a single English dictionary. You're going to completely miss concepts that only exist in French, Japanese, or Swahili.
That's a great analogy. So to fix this massive blind spot, Xu Kai and his team developed an algorithm and a method called pan blocks.
Pan blocks.
Yeah.
How does that work step by step?
Instead of using one reference, they took 21 distinct Brassica rapa genomes from across the globe. They started with genome 1, then layered genome 2 on top of it.
It's like stacking transparent maps.
Exactly. They extracted all the matching blocks of DNA called synthetic blocks and merged them. Then they systematically repeated this layering process for all 21 genomes, filtering out redundant sequences along the way.
So they essentially compiled a master lexicon. They took every biological language spoken by these plants, merged them, and deleted the duplicate words, leaving behind a complete encyclopedia of every possible structural variation.
Yes, and the pan blocks allowed them to capture nearly 100% of the genetic variation. They ended up with a map uncovering over 24,000 genes that were totally invisible when scientists were only looking at the old Chifu blueprint.
Okay, let's anchor this concept with a quick recap before we move on.
Yeah.
So the Brassica family is built on this triangle of U, combining base genomes to create complex hybrids. But our understanding was severely stunted, because relying on a single reference genome hid up to 2/3 of the plant's DNA.
Right.
And the pan block technique fixed this by weaving 21 different genomes into one master map. Which makes you wonder, and I'm asking you, the listener, directly, if you could only see 33% of a puzzle, how wildly inaccurate would your guess of the final picture be? And more importantly, how much of our current understanding of human medicine or biology is limited right now by this exact same single reference bias?
That is exactly the question keeping a lot of geneticists away at night right now.
So now that the team finally had this complete, uncompromised pan blot map, they unleashed it on 3,330 different plant samples. What did this supermap reveal about where these vegetables actually came from? I'm assuming they found some pristine, wild ancestor growing on a remote mountain.
Actually, the data points in a very different direction. By tracing the root of the phylogenetic tree, the absolute origin point of the A genome zeroes in on Central and West Asia.
Okay.
But it wasn't a prized wild vegetable. The genetic and archaeological evidence suggests Brassica rapa began its relationship with humans as an invasive weed growing in ancient winter wheat fields.
A weed? Wait, bread wheat goes back something like 8,500 to 9,000 years in Mesopotamia. You're saying the ancestor of bok choy and mustard was just the annoying brush popping up between the wheat stalks.
Yeah, it was an agricultural pest, but it survived because of a brilliant evolutionary trick. It had a very similar growth cycle to winter wheat.
Oh, sneaky.
Very. When early farmers watered and harvested their wheat, they inadvertently watered and harvested the Brassica wheat alongside it. And over time, those ancient Mesopotamians realized that tiny seeds of this weed were actually packed with essential fats.
I'm guessing that is where the oilseed varieties come into play.
Exactly. And the genetic data perfectly confirms this timeline. When we look at the sheer variety of genetic mutations, what biologists call nucleotide diversity, the oilseed varieties of Brassica rapa have the highest diversity by far.
And high diversity means it's older, right?
Right. In genetics, the highest diversity almost always points to the oldest population because they've had more time to mutate. So humans started crushing these weed seeds for oil long before they ever thought about eating the leaves.
That is wild. Human civilization was basically built on a side hustle crop. We were focused on farming wheat, this weed hitched a ride, and we eventually realized it was a free source of cooking oil. So how did a localized Mesopotamian weed conquer the globe?
The researchers tracked 3 distinct migration routes across Eurasia.
Let's break them down.
First was the South Asian route, moving downward to create Indian sarsen, which is a crucial oil crop. Second was the European route, moving westward toward the Mediterranean, which gave us the European turnip and leafy rapini.
And the third route?
The third route is the most dramatic expansion, the East Asian route. The seeds traveled eastward along the prehistoric Silk Road.
Oh, wow.
Yeah, they passed through harsh, varied climates, moving through trading hubs like Gansu Province in northwest China. Once the plant hit East Asia, its genetic diversity just exploded. It branched into northern China to become Chinese cabbage, southern China to become pak choi, and eventually crossed the water to Japan to become mizuna.
Okay, let's do a quick recap for the listener here. If we map this out mentally, the ancestor of our cabbage and mustard was an opportunistic weed hitching a ride in 9,000-year-old Mesopotamian wheat fields. From there, human trade pushed it south to India, west to Europe, and east along the Silk Road, forcing it to morph into everything from root vegetables to leafy greens just to survive.
Spot on.
Now, next time you are eating a salad, look at those leaves and consider the thousands of miles they traveled. It makes you ask, what invasive weeds currently growing in the cracks of our sidewalks might actually contain the genetic traits needed to be the supercrops of the future?
The adaptability of weeds is unparalleled, which naturally leads us to the mystery of how these simple plants hybridized to survive those brutal Silk Road journeys.
Right.
Remember the tetraploids from the triangle of Brassica napus, the rapeseed, and Brassica juncea, the mustard?
Right, the complex plants with 4 sets of chromosomes. If the original weed was spreading in all directions, how do we know exactly when and where it smashed its genome together with other species to create canola and mustard?
For a long time, we didn't. But the pan-block analysis uncovered a literal structural smoking gun at the very end of a specific chromosome called A03. The researchers found an ancient inversion.
An inversion. Let me see if I grasp the mechanics of this. Is it like someone took a paragraph in the plant's genetic instruction manual, cut it out with scissors, and pasted it back into the genome completely upside down?
Exactly.
So the letters are all still there, but because they are facing the wrong way, the cellular machinery reads them differently.
That is the perfect way to visualize it. This specific upside-down paragraph on chromosome A03 created a massive fork in the evolutionary road. You end up with 2 deeply diverged lineages. You have the original paragraph, which we call HapXC04, and you have the newer flipped paragraph called HapCFV4.
Which version won out in nature?
The flipped version absolutely dominated. The researchers found that 93% of all the Brassica rapa plants they tested carry the flipped sequence. It is nearly universal in East Asia.
Wait, if the flipped version is so dominant in Asia, but not Europe, that implies the mutation gave the plant a massive survival advantage as it traveled east.
Exactly. As the plant moved along the Silk Road into harsher, colder eastern environments, the inverted DNA sequence provided some sort of adaptive edge. The environment selected for it, and the trait swept through the population. But the real breakthrough is how this inversion acts as a genetic timestamp for our tetraploid hybrids.
How does an upside-down paragraph tell time?
Because of who carries it. When the team looked at Brassica napus, the rapeseed, every single genome maintained the original paragraph. None of them had the flipped version.
Oh, I see. That means rapeseed had to have formed its 4-chromosome hybrid structure before the upside-down mutation ever happened, or at least before it became widespread.
You nailed it. It proves rapeseed diverged incredibly early in the plant's history, likely in the West, before the European turnip had even fully taken shape.
Wow.
But when they looked at the other hybrid, Brassica juncea or mustard...
Let me guess. 100% of the mustard plants carry the newer flipped paragraph.
100%. Which proves mustard was formed much later, specifically during that eastward spread along the Silk Road, after the DNA had already mutated to survive the journey.
Okay, let's synthesize this timeline for everyone. The A genome underwent a massive structural mutation, an inversion that flipped a sequence of DNA backward. Because rapeseed only carries the original sequence, we know it formed very early in Western history. Because mustard only carries the flipped sequence, we know it formed later as traders moved the plant east.
Perfectly summarized.
It is incredible that our DNA carries these timestamps of ancient migrations. It makes you wonder, what hidden journeys and ancient environmental adaptations are encoded in your own genome, just waiting for a map detailed enough to read them.
Human history is entirely written in our cells, just as the Silk Road is written in these plants. But, spreading across continents and smashing genomes together created a dangerous biological problem. Well, if these plants are constantly reproducing in tight agricultural clusters, how do they avoid catastrophic inbreeding?
Oh, right. Because if a plant just pollinates itself over and over, you hit a genetic dead end. You lose the adaptability that allowed it to survive the journey from Mesopotamia to Japan in the first place. I know animals avoid this by, moving around, but plants are rooted to the ground.
Right. To solve this, the Brassica family utilizes an incredibly complex defense mechanism called a self-incompatibility system, or SI.
So it's basically a chemical filter that lets the plant swipe left on its own pollen.
Essentially, yes. At the molecular level, this filter operates in a region of the DNA called the S-locus. Inside this region are two crucial genes. You have the SCR gene, which acts as the male identity marker on the pollen, and you have the SRK gene, which acts as the female identity marker on the plant's pistil. They function exactly like a lock and a key.
Let me get this straight. If the plant's own male pollen lands on its own female pistil, the key matches the lock, and the plant actively rejects it to prevent inbreeding.
Right. It stops fertilization dead in its tracks. But there is a massive vulnerability in this system.
What's the flaw?
During reproduction, organisms undergo a natural process called genetic recombination. They literally shuffle their DNA, crossing over strands from mother and father to create variation in the next generation.
So if the cellular machinery accidentally shuffles the DNA right in the middle of that S-locus, it would separate the SCR key from the SRK lock.
Exactly.
The whole system would break down, and the plant would suddenly be able to inbreed. How does the plant stop the cellular machinery from shuffling that specific spot? Do they just keep the lock and key on totally separate chromosomes?
They actually have to be inherited together to work. The solution they found is what the researchers call a genomic safe, and this is where the pan-block mapping technique proved its worth yet again. When they finally got a clear look at the S-locus, they found it was absolutely packed with transposable elements, or TEs.
Wait, transposable elements are those jumping genes?
Yeah.
Like the bits of junk DNA that just copy and paste themselves randomly around the genome.
Yes, jumping genes. The plant has essentially flooded the S-locus with them. They make up roughly 43 to 55% of the entire DNA block in that region. It is totally saturated with chaotic clutter.
This is fascinating. So the plant intentionally surrounds its most vulnerable, important genes with a massive moat of genetic garbage.
Basically.
It is like packing a fragile glass vase in thousands of wildly different shapes of packing peanuts. The replication machinery gets in there, gets completely tangled in the junk, and physically cannot perform the crossover.
That analogy is spot on. The jumping genes act as an architectural barrier. And what makes it brilliant is that the clutter isn't uniform. The jumping genes form a unique, chaotic barcode for every different variation of the S-locus. One variation might be packed with a type of transposon called gypsy, while another is packed with copia transposons.
Because the sequences of junk DNA are completely mismatched shapes and sizes, the two strands of DNA physically cannot align with each other to swap material.
The structural chaos literally paralyzes the recombination process in that specific area. It creates a non-recombining region. By using chaos, the plant maintains perfect order, keeping the lock and key bound together across millions of years of evolution.
Okay, let's do one final wrap-up of this mechanism for the audience. To prevent inbreeding, the plants rely on a strict lock and key gene system. To prevent those two genes from being accidentally ripped apart during natural DNA shuffling, the plant surrounds them with a chaotic moat of jumping genes, creating a structural barcode that is impossible to tamper with. It is a literal genomic safe.
It's a marvel of evolution.
It really is. Think about what that tells us about biology, you know?
Oh, yeah.
Evolution built a tamper-proof vault using bits of rogue DNA. What does it tell you about the natural world that sometimes absolute chaos and junk are exactly what is required to maintain order?
It forces us to completely rethink the genome. It is not just a clean, linear set of instructions. It is a dynamic physical landscape where even the supposed clutter serves a critical, life-saving architectural purpose.
As we pull all these threads together, it becomes clear why this Nature Genetics paper is so monumental. By abandoning the blind spot of a single reference genome and building pan blocks out of 3,330 different plants, these scientists didn't just map the DNA of a few random vegetables.
They successfully mapped human history, they traced ancient agricultural practices in Mesopotamia, they followed trade routes along the Silk Road, and they decoded the brilliant, chaotic defense mechanisms that allowed these plants to survive alongside us for 9,000 years.
It is truly staggering. The next time you are standing in that produce aisle, deciding between canola oil and mustard, or choosing between bok choy and a turnip, I want you to remember that genetic hall of mirrors. You aren't just looking at isolated ingredients for your dinner. You are interacting with an ancient Silk Road traveler that survived the millennia by constantly rewriting its own genetic barcode.
And it serves as a powerful reminder to the scientific community. If you want to understand the true mechanics of life, whether in crops or in humans, you cannot rely on a single blueprint. You have to embrace the full, messy diversity of the population.
I couldn't agree more. And I want to leave you with one last thought to mull over. We have spent centuries, perhaps millennia, trying to eradicate weeds to protect our pristine, orderly crops, only to discover through deep genomic mapping that one of our most important, globally sustaining crop families was the weed.
Right.
It makes you wonder, as shifting climates force us to radically rethink global agriculture and survival, are the incredibly resilient, highly adaptable weeds currently growing in the cracks of our sidewalks actually the ultimate genetic blueprint for our future food supply?
That sounds like the perfect mystery for another deep dive.